Okay. Well, thanks for inviting me. When I get invited to these talks, I'm really, some people like to talk about their grand theory that they've worked on for 15 years, and that's a recipe to basically put everybody to sleep. So this is going to be a little bit of a mix. There's not going to be any grand theory. And we're going to go through a bunch of results that I just thought I put them together because I thought they'd be interesting. And a lot of times I don't have the full answers for these things I'm going to present, some of the data. So they're going to be more questions than really answers. We're going to have some answers, or at least we're going to try to give some answers. But when I think back on what are the talks I enjoyed the most, I always come out, I saw this talk, and really there were not many answers. They asked great questions, and I realized that's a great way to go around the world. You just ask questions, you never answer them, and people are happy. So anyway, so we're going to jump back and forth a little bit. Please do interrupt with questions. I'm going to try to because I know some people are outside the field of vision science. We have sort of a mixed crowd. I'm going to try to explain things as well as I can, but do interrupt. And of course, the title doesn't really reflect everything that we're going to be covering. If we do cover, we get to cover everything. But that was one attempt for a title. So the starting point is really what I had in the abstract. Oh, first, another thing. When you give these talks, you're allowed to bring guest data. So I don't know if you guys have that tradition, but I'm just putting that. You're giving So I have some guest data from some of my colleagues, which I think you sort of improve the story. And here are basically all the people that helped out and did most of the work that I'm going to be presenting, and I'll mention their names as we go through. Otherwise, all of this stuff wouldn't be there. Ooh! So that was good. That's it. Okay, well, fortunately, you're going to believe, but I have a bad reputation with these things happening. So I am practiced at this. Actually, one time at NYU, at a job talk, something like that happened with the projector. I had to give my whole job without the slides. But this actually looks better. It's going to come up. But that is unusual. Okay, so let me start. Let me give you where is the starting point. So our starting point, while this comes up and I log in, is I don't know if anybody had actually listened to NPR this morning. There was actually a piece, a friend of mine told me about it, on guns, tumors, and the limits of the human eye. So the starting point of these tasks that I think I talked about in the abstract, challenge these tasks like finding a tumor in a medical image, or finding a weapon in an X-ray image, a baggage X-ray, or even finding something in a satellite image. All these are very challenging tasks. And you might imagine that if you just have either normal vision or corrected vision, if you just go through some training, you should be able to do these tasks fairly well. So by the end of this talk, I hope to give you some evidence that might not be the case. That there are actually huge differences in how we actually perform at these. We tend to refer them as life-critical perceptual tasks, or any challenging task. Some people are better, some people are worse, and we're just starting to starting to understand what are the reasons. So I'm going to try to convince you of that. And in the first part of the talk, I'm also going to show you not only that people actually do perform differently at these tasks, but how they actually explore an image, where they point their eyes. So where the center of the eye goes as they actually do these tasks, systematically vary across people. And even for the most elementary tasks, it's actually identifying. So just take out all these specialized tasks. Let's go for something basic. I put a face up and you have to identify who it is. I'm going to show you some data that actually shows there are systematic differences across people, how they look at these faces. And we're trying to understand why why are these differences, why are they there? Then in the second part of the talk, I'm going to actually try to do, we're not going to just look at behavior, but we're going to try to see if we can actually measure neural activity from humans and attempt, and obviously this is going to be non-invasive, indirect measures of neural activity, because we can't, because for the most part we can't do direct measurements of neural activity. And we're going to try to see from the neural activity if we can actually predict how well somebody's going to do in this task. So that's sort of a challenging thing. But there's a lot of groups in the country and the world that are actually trying to do these sorts of things. Okay, is everybody trying to guess what that is? It's like everybody's looking at the picture. Any guesses where that picture is?
No, I'm wondering what Windows came up out of
Oh, is that what you're wondering? Boy, you spend too much time. It's too much academic life there. It's like, why you have that? And you say, that is the reason it crashed. And you're probably right. We just had a conversation about boot camp, and I says, well, it has its things, but overall it works. But it's never done that, actually. So the blue screen is not something I get often. So no guesses on where the I still didn't get a guess of where the picture is from. If somebody is not thinking about why you have a Mac, any bets? Well, it's not Argentina. That's a good bet. That's a good bet. It's Nepal? No, that's a little bit better, but it's not there. Anything else? Oh no. Okay, Yemen. Yeah. Well, a few years back, it was good that I went then. Now it would be a little bit more iffy. Okay, so we're back here, and interesting. Oh, there we go. Okay, there we go. So here are the life-critical perceptual tasks, challenging tasks. Here we go. Security, satellite imagery, breast imaging here, X-ray mammogram, computed tomography, CT. So these are the sort of tasks that I'm going to be talking about, because the stuff I'm going to be talking about is relevant to these sorts of tasks, because it's important that we have people doing these things and they're good at it. Okay, so I said that I was going to try to convince you that people have different abilities at doing these things. So the first thing you can do is just bring people to the lab. These are undergrads, so it's too expensive to bring the doctors in. Let's start with the easiest thing you could start. It's a very artificial task, I know, but we'll start with something like that. So in a screen, so this is what these observers are looking for, and this is, can everybody see a little bright spot there? That's a luminance target, and it's not that bright in the task. It's embedded in this, which is noise, white noise. Each pixel is randomly perturbed with luminance, and that obscures it sometimes, so sometimes it's very hard to see. And half the time, so the person is sitting there and an image comes up, and half the times that target is there, half the times the target is not there. There is just noise. Now the noise sometimes, because it's a random process, happens to make something that looks like the target, and people will incorrectly say the target is there. So they're making these decisions, yes or no, and they sit for like a week, not all day a week, but 2 hours a day. So, we collect about 2,000 trials. So a trial, each of these individual decisions that has independent noise and the target could be that, I'm going to be referring to that as a trial. And if you measure performance for these, in this case 20 observers, I'm going to have this metric here, which is a metric that tries to compensate for the fact that some people are more propense to say yes than no, and so on. And chance is 0.5, and the highest performance you have is 1. That's the best you can do. And you can see, and I've ordered them here, so it's not like I just collected them like that and by chance. I've ordered them, but you can see there's a pretty significant variation on how people do this simple task. And I know what you're thinking. You're saying, well, yeah, but this is a very simple task. The subjects are not experts because this is, if you bring them to the lab, they've practiced before they started, but this is really an artificial task. So then we thought, well, why don't we try them at something that supposedly they do every day, like identifying faces. So now they come to the lab, other people, not the same 20, and they press a button, and then an image comes up, and if you can barely see it, there's a face here. Does everybody see? There's a very faint face. This also has noise added, white noise, and there's going to be reasons why that's going to be important. There's something called a mask, and that's just presented so that we interrupt the after image on people's visual system. And then this image comes up with 10 faces. Now they've learned these faces, so it's not like I just started them there and they've never seen these faces. So they've practiced this before, and they've learned these faces. These are not their relatives or friends, they're just 10 faces. And then they click on the face that was presented here. Each face has equal probability, so a tenth of probability of being sampled on each trial. and then they make a decision, and we record how well they can identify faces. And they do this 500 trials, so 500 decisions. So here we go. So chance here is 10%, and the best you could do is 100% or 1.0 and 0.1. And at this time I didn't order them, and they have error bars because they're doing fewer trials, so we want to put error bars now. But still, huge differences. You know, some people they're pretty good at this. They're about 80% recognizing faces, and there's a lot of people that are like about 30%. So, you know, they're not very good. Now, I know what you're thinking again. You're saying, yeah, okay, but, you know, how stable is this? How replicable? If you bring them, you know, if you tell me you bring them next month, and this person becomes really bad and this person becomes really good, okay, there is variability, but maybe this source of variability has to do with the fact that they had a bad day. And part of the difficulty in these studies is really trying to figure out, because I can't control who went to, you know, if somebody partied all night and came and do the task, and they're, you know, or they didn't sleep, or they had a midterm, so it's difficult. But we want to know whether these differences are really replicable. So this is Matt Peterson's work. So these are 5 people, and we're in the process of bringing them back. Some of these people came back. So this is the original data I just showed you, and this is the retest, which was done between 5 months and a year later. Same 512. And there are differences, of course. And some people got better. Probably they learned a little bit. But overall, you pretty say, you know, the good people are good. The bad people are still not so good. So there's, huh? Everybody got a little bit better. So that means that they're still learning. They were still learning. Even though we try to give them some practice, they're still getting a little bit better. Yeah. Yeah, it's the same 10 faces, and they go through, once they come back, they go through a little familiarity to just get a little refresher, you know. So you're not like just thrown there and like, what are you talking about? These 10 faces I haven't seen in a while. So you get a little refresher, and then you start again. So it's the same 10 faces. And but then that's interesting. What if you do other faces? So in general, we don't think there's nothing. We don't think, but that's another thing. We don't think there's anything specific about these 10 faces. So it's not like you're just really good at, you know, recognizing Arnold Schwarzenegger and Stallone, but then with, but you know, that's what we think. But like I said, we don't. There's a lot of questions here. Okay, but at least I could, you know, maybe you're semi-convinced that you know, there's something here. That there's some individual differences that are actually replicable, even in a pretty, you know, basic task. Now, what you probably some of you are thinking, or you should be thinking, because this is what we thought, and is like, well, yes, but maybe the only reason, you know, the reason this person does better than this person is just this person is a very responsible undergraduate that takes this seriously, and this is really, you know, they're not motivated. Really, I mean, they don't really care. I mean, if I don't care, I just don't, you know, what, you know, psych, experiment, who cares? You know? I mean, I come and I just, so this is very possible. And so what we try to do sometimes is like, well, what if we actually give them some money for each time, each trial they get it right, like 10 cents, and then you get them motivated. But the problem is, like, you could always say, well, 10 cents is not much. So we have some experiments where, you know, 10 cents is not much. So we can up it to a quarter. But then you can see, well, it's a quarter per trial. Yeah, it still makes you, but, you know, that could get very expensive very rapidly, and you're never convinced unless you give them a lot of cash that they're truly motivated. So to give you an example that this is not a motivation problem, I'm going to bring Craig Beam's data. So here's an example where so here is subjects are not motivated enough. This is a lab effect, and this is why we get systematic differences in how people do these perceptual tasks. Now, to convince you that, in fact, this is not a problem, this is not a motivation issue, I mean, this is what we think, I'll bring some of the data from my colleague at University of Illinois, Chicago, Craig Beam, where he actually measured performance of actual mammographers, radiologists, looking at finding tumors in X-ray mammograms. And he had a database of 135 cases that he actually shipped across the country and had different radiologists, all these different radiologists across the country read exactly the same images. Now, you could always say that, you know, that they're not motivated, but, you know, it's a little bit of an issue. Now, at that point, you know, this is a task that they're actually, that's their profession, that they should be motivated. There's always that comeback. But you would imagine that you know, that if it was a motivation issue, now the variability in performance for this task should be reduced. Now this made the when he published this data I'm going to show you, this made the headlines in 2003. It was actually even in USA Today. When you hit USA Today, you're just that's when you know you have something important, you know. So here's a radiologist, and chance is 0.5 and 1. And you know, there's a good, you know, look at this person. We don't want this to be your mom's or your wife's radiologist, definitely. So there is a striking variability. Furthermore, in this paper, Craig Beam concluded that the variability in diagnostic accuracy reading mammograms is that variability is the main source of variability is human performance, and it's much greater than the variability in diagnostic accuracy due to equipment and whether it's calibrated right or wrong. Because, you know, we all spend, you know, all these X-ray physicists spend time calibrating these things, and of course there's some clinics that have better equipment than others. He concluded that the main source of diagnostic accuracy variation, this is much larger than what you see in the equipment. At least that was his conclusion. So that was, you know, that's sort of worrisome. So that means that, you know, maybe there's something here to understand, and obviously we will want this person to get better. And do we know whether this person can get better? I don't know. We don't have the answer to that yet. Yeah.
Were these people checked out in a regular eye test?
Most of them have normal or corrected vision. I don't think these people were actually. Perhaps these were. I mean, I don't know. I have to go by a paper. I doubt it that they're actually. During this study, they were actually basically measured. But most radiologists have to basically, you know, when you actually operate, you know, you have to have corrected vision. That's sort of a basic thing. If you're not like, although, you know, people also claim whether that has an impact, what's the impact of that or not, depending obviously on your correction. But
it's very indirect. I mean, they have corrected vision. Nobody has absolutely
corrected vision. Well, I don't think anybody has measured the variations, but I would actually tell you that those are smaller than what you see here. And on top of that, a lot of these corrections, you lose some of the high frequencies, some of the high frequency information, and a lot of these targets actually happen to be a little bit more low frequency. So, but that's a good question. Can you actually, you know, quantifying how much is it? But there's much less than this. That's what my take is, and then, you know, if there is any. So the variation due to the fact that it's not an optics thing. That's basically the issue. I mean, the other thing to know is if it was just an optics thing, you can see how people improve through time when they do this task, and the performance improvement due to learning is huge, even though, you know, the optics is always the same. So these are really things having to do, for the most part, post optics. These have things to do with how, you know, your and I guess. You know what is a good answer to your stuff? The second part of the talk, where I'm trying to actually connect neural activity to performance. And if it was just an optics thing, well, you could say that that's also impacting the neural activity in some way. But yeah, I don't think that's it. But we can talk about it more later if you have.
Well, just one quick remark.
Yeah.
Namely that U0 is set at 0.7.
Yeah, 0.5 is chance.
The actual variation is percentage-wise, not different.
So 0.5 is chance. Yeah, this graph happens to be from Beam. It happens to start at 0.7. So yeah, so I guess if you wanted to compare, I mean, we could look at it, but you maybe put it at the same axis. But some of the other tasks, chance is at 0.1. So it's, you know, so there's variation, but it's hard to do some of these comparisons because some have chance starts at different starting points. Okay. So hopefully by now you have an idea that there are these variations that are real. Now, yes, you have a question in the back.
Well, it's the same question of how much is local variability and how much is global variability. So this was 10%. So what do we compare 10% to? It would be helpful here too to have variability, from one day to another.
Right. So that's what I showed. That's why I showed you the control experiment where I brought people. That's sort of, if we go back here. Now, doing this is a big NIH grant that takes. So to do a second measurement would be expensive, but we can do this sort of stuff, where I can bring people back for retest, and you can actually see sort of this. Again, chance here is 0.1, and you can see that there is variation. This is nearly a year later, some of these people. Some of these are 5 months later. And there is some variation, but the variation across repeats is less than the variation across observers. So that's sort of It'd be great to get the doctors back, for sure, but expensive. Okay, so now not only is performance difference for these people, but actually how they explore the scene. Now, I'll give you like a one sentence on that. I think most of you know, maybe know about vision science. I know there's people from psychology, but those that are not familiar, I think some of you know that the center of the eye has high resolution, and towards the periphery you have sort of lower resolution processing. It has to do both with things having to do in the retina, the density of receptors, but also the sort of preferential processing that the cortex gives to the fovea. And that's why we move our eyes, because we want to point our fovea to points of interest. And what we wanted to see is how doctors and people basically move their eyes. And this is another, so what you see here is another slide from a colleague of mine at University of Pittsburgh. And this is, these are, now just so you're not confused, these are two x-rays from the same patient, but they're taken from different views. And where there's lots of stuff here, but where the red circle is, that is where the actual tumor is, but it's from two different views. These are exactly the same images as those. Okay, so that, I told you that's where the tumor is, and you'd say, what? There is nothing there. In fact, there is. Okay, but it's just, and the reason we know is somebody did a biopsy, and that's how we know this is that there is actually a tumor there, and that's where it is, and it's the same here. That's the same two images. Now, what you see here with a device called an eye tracker, which most of these devices are just infrared video based, meaning that you just have a source that's infrared, and you collect on a detector the image of the eye, the pupil, and then a corneal reflection, and then you send that to a computer and you analyze it, and you can actually determine where somebody's looking at. And those little circles is where people are actually looking at. These big circles that Claudia put, these they shouldn't be. I mean, they just get on the way, but it's just to illustrate that there are clusters of fixations. But each fixation is this, there. And this is one person, one radiologist, and this is another radiologist. So if you're going to get one thing from this talk, is how to pick your radiologist from their eye movements. So I'm going to give it to you. Which radiologist would you want? You want this radiologist on the left. So this is the same radiologist looking at the two views. That's their eye movements. And this is another radiologist looking at the same images, but the two views. So anybody? So people that like your left. People that like the left. Nobody wants the radiologist. The left. Okay. People that want the right. Okay. And a lot of people don't want. They just don't want to get it. I saw. You're going left. Okay. Brian. So we have a visitor from Stanford. Okay. So, surprise, perhaps. This is the novice, and this is the expert. So this is a case where there's a difference. Huh? Thank God? Okay. Okay, we'll get a thank God. So what's going on here? This is a case where people have studied where you can actually really understand what's going on. Novices, typically, they're just looking all over the place. And experts really just do fewer eye movements and then make the decision faster. Now, some people, when I presented this at SPIE a few weeks ago, Stephen Palmer says, well, what about, he doesn't even look at the tumor. The expert. And I was like, yeah, that's a good point. But then again, if you're really good, you might not even, you could actually just look at right close to the fovea. Just, so you're like, you fixate slow and they say, yeah, tumor, doing, moving next. I don't even have to look at it. So that's a good point. But, that's what it is. I mean, Claudia can send me more, and this is documented many, many times. So this is a case where we know why people have fewer eye movements because, they're experts.
What is the expert better?
Expert is better. So typically, not always, but it's a distribution. Like everything in life, there are distributions. But in this case, the expert is better than this novice. But in general, if you tell me, are experts better? Experts being more training, more years of residence, it's a distribution. Experts are the mean, or the medium is typically better, but a lousy expert is better. This case, this better. The expert, yeah. I think that is, I don't think anybody has looked at that, or I haven't seen that reported, and if I haven't seen reported, it's because that's a much trickier thing. I think probably once you get experience, your eye movements are okay, but there are other things that might determine whether you're going to, that are not perhaps captured by the eye movements. But we'll come back to medical imaging because I have some guest data from your former graduate student, Steve Engel, which actually is about this. Okay, so let's, this is a case, differences in eye movements. What do we go back to, but let's go to something basic, like identifying faces. I already talked about the people's performance is different. Now, where do people look when they do that task? So if you remember that task, one thing I didn't tell you that we did is we actually had people on an eye tracker, so we could actually look where they were actually looking at within the face when they're doing this task. Now this is presented very rapidly, so you just have time for about one eye movement or two. And you start, your initial point of fixation is at a random location outside of the image. So you could start here, 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 and then the face comes in, and the test image, and then you make your eye movement, and then you do your decision. So we tried to see, we have 500 trials, so we would have 500 points of where people look at when they do these tasks. So this is one person, BB. And this is fairly common. So those are the little 500 points. That's the end point of the first fixation. And now you'd say, well, for which face? There were 10 faces, And Matt looked at the different faces, and he didn't see any systematic differences. So what we've put here in the background is an average face. And these faces were a priori sort of scaled to actually sort of have the same area and so on. So this worked. And these are, so this person tends to, the centroid is about there. So now we had 25 people. So now if we actually collapse all these 500 points into centroid and just show you that 1 point per subject. So now this is 1 point. Each of these points comes from 500 points. And there are two things that are interesting here. The first thing, people have seen this all the time. This is, they tend to, for the most part, they tend to be close to the eyes. And sort of the wisdom has been, there's a vast literature that we're just trying to go through. And some of the reasons some people think that's, people in social psychology think it's sort of like a cultural thing. And then other people say, well, yeah, the eyes make sense because they sort of have the information. But there hasn't been anything really rigorous quantifying that the eyes really have good information if you want to identify somebody. So what Matt did is he tried to have an objective metric to quantify the different amount of information that you have in different parts of the face. And some of you, so one thing you can do to try to do that is to use something called, probably most of you are not familiar, something called the Bayesian ideal observer that calculates, and this is why we actually put noise in the image, because given that you have these images and you have this noise that you've added to the images, and you know the statistics of the noise, you can actually posit what is the optimal algorithm to do this task based on just mathematical statistics and probability. Yeah?
Is the dispersion around any one of those individual points compared to the dispersion of the different values? They're trying to figure out is that dispersion.
I'm going to show you, in the next few slides, I think I'm going to answer some of that. And then if you're not satisfied, you can go at it again.
There seems to be a noticeable bias.
I know, everybody asked that, and everybody asked us, and it's another one, maybe Brian. So we've been trying to, there's a lot of hand waving about why that is having to do with hemispheric lateralization and stuff like that, and that's about as much I know, but everybody gets it. Everybody tends to, everybody sees, all the labs get that people tend to look a little bit more to the, not everybody, but on average, they sort of look a little bit more to the left eye than the right eye. And there are reasons, but it's sort of a little bit of a mystery. People try to link it with, it's not, I think I've asked Matt to go research whether it has to do with handedness, and yeah, I know, it's like the obvious one. Is it right-handed and left-handed? But no, I don't think it has to do that. It's always left, whether you're right-handed and left-handed, and there is some explanations, but they're not fantastic. Say that again? Ah, there you go. Yeah, that's a good one. Our Brits, yeah, that's a good paper. Brits look right. I like that title. Publish it in the Royal, what is it? The Royal Society. They'll like it. Okay, so what I was telling you that there's this sort of mathematical construct that comes from mathematical statistics, and also people have used in electrical engineering. And it's so you implement this model in a computer, and it gives you the best possible performance for a task. And given that we have this noise that we've added, we can calculate that. But critically, what Matt did is he actually fed to this model little bits and pieces of the image. So he just, oh, by the way, he just didn't do it for this 10 images. He did it for a database of 1,000 images that we actually collected, and it was a little bit painful, but we got 1,000 images that were all cropped and normalized and so on. And he would he ran a huge simulation where he get 10 faces at a time, and he was trying to, because you want to quantify how much information is there in different parts of the face for sort of a population of images, not just the 10 images that you have. When he did that, he got this. So this is a performance map, sort of proportion correct, of the Bayesian ideal observer as a function for little bits and parts of the face, sort of a heat map. And you can see that according to this metric, indeed, the eyes do have most of the information to discriminate or to identify. And of course, this is going to be task specific. So if you have to do a task which is smile versus not smile, you'd imagine now the smile is where the information is. And then you say, well, did you measure eye movements? Oh, yeah, no, that's what we want to do in the future. But this is sort of a nice rigorous thing that we can actually show that using sort of a metric, we can actually quantify the information, and that sort of makes sense. But that was sort of the obvious answer. The more kind of complicated answer, like you were saying, is like, there's somebody, okay, yeah, they go to the eyes, but no, hey, that person goes to the nose. And then we could say, well, is this a real variability, or is this reproducible? That's the other thing. So here we go. So here I'm going to show you somebody that goes to the eye, BB, and then JG that tends to go to the nose. And that's all of the 500 ones. Now I know what your next question is. Again, is this reproducible? You know, come on, yes, you showed me in one sitting they have different things, but are they, you know, if I bring them back a year later, are they going to look at the same place? Okay, let's look at it. So here's JG, July 2008, February 2009. Pretty good, no? Not bad. So, and then we have, now I put you two other people, BB, original, retest, and then another sort of nose person, VH, I know, retest. We were impressed by that. We were like, wow.
So the question is whether the nose person do a lot worse.
Ah, good question, Tony. That's what we thought. That's where we're going. But before that, we thought, well, maybe it has to do, you know, what about, is this, you know, because we're just doing 10 faces. You know, does this really generalize to the world, to other faces? I mean, I know you're thinking about that. Yep.
I can't tell from the initials, but is there any gender differences?
I think we're able to disclose those because, you know, I know you're in theoretical physics, so you guys don't know about human subject committees. But I think genders we can disclose. So they're both here, female, and we're trying to do some analysis across to gender. And we haven't really got to, or we probably have more females than males. So, but that's something. It'd be nice to see if there's some gender differences across, but we haven't looked at that yet. Here the two, both females. That's what I get from Matt. So this is okay. But yes.
You have a wrong noise model. Does your Bayesian observer have a different part of the face look like?
Well, I think it's a tricky question because we don't. The images, we create the noise. So we don't have the images, but okay, so the noise we know. What you're saying is a really important thing, which is, what if the noise that the brain adds has a different statistics than the white noise? Now the Bayesian ideal observer, taking into consideration the sort of statistics of the brain noise, that is different. And that is a big sort of issue with all these approaches. And what we tend to think is that just to convince ourselves and feel better, is that with the white noise you're overwhelming the system, and so the white noise is sort of the big part, and if there is some brain noise that has a different sort of spectral density, then it's overwhelmed, so it's a good approximation, but it's an issue. Yeah. So back to this, we wanted to try this with other faces. So what we did is we had Matt have famous people. And the nice thing about famous people is, well, the thing is they're not black and white like the other ones. We're not going to put noise, because some people would say, well, you had noise in the image. People are not going with white noise in the real world. True. So we put 100 faces up, and you had to recognize them. And they were scaled again, and so on. Everybody should recognize these. If you don't recognize these, come talk to me. I'm in the psychology department. Okay. So there are fewer of these, 100. So here, BB and VH, there's more spread here, true, but pretty impressive. There is so we put the famous faces and they're looking at the same spot. So it's a consistent sort of pattern that people are doing. This one is more spread out, and I have about 6 or more of these. So I'm just showing you 2. But it's pretty consistent. Somebody going to the eyes, go to the eyes, and the one in the nose, it's a little bit more spread out for the famous people, but they go there. So then, I came back from this meeting. I said, Matt, I know I give Matt a lot of headaches. What if we inverted the faces? I know, it's like, because once you start with this, and then, then he ran, so then people came and reran it, and they had to recognize the faces, but inverted. And let's see, so you're like, so this is the eye person, and this is everything. The only thing is, it's now they still on the left. That's sort of interesting. So they don't go here. They actually go, so they're actually looking at a different eye. Because they're still going to the left side, but now that's that eye. And then the nose person here, that's looking there at the inverted faces. So there's something there that's interesting, a persistent difference in how they explore these. Now to Tony's question, why are they doing it? That's what our question was. So why is this a random thing? You just start doing this and you just stick to it? They have sort of, they can kind of put a second one, but that wouldn't get it, so they have one. We haven't got there, but that's sort of, if that's a systematic thing. But what I'm going to show you might motivate some of those experiments you're thinking about. So when I told you about this experiment, and I told you that they started here and they made an eye movement, we also had a separate condition they actually ran that I had not told you about, where they actually had to do the task but maintaining fixation. They could just, the cross comes up, so the cross comes up here. You have to maintain fixation and do the task. You cannot move your eyes. If you move your eyes, the eye tracker detects that, automatically aborts the trial. Okay, so this is so you and what we did is we manipulated fixation. Now here's and then we measured performance as a function of fixation. And then the idea was like, well, if people move to the eyes, does that mean that when you actually force them to fixate, perhaps that's the hot spot where they actually do good? And this is what we found. So here's the eye person, and this is sort of the performance map. And there's some interpolation here and so on, because obviously we didn't measure everything in a millimeter. There's so many trials we can do. But you could tend to think that, that sort of the hot spot is around there, and then this nose person, that's their performance map in the fixation. So what we're seeing is, that is close to where they perform the best when they're forced to fixate. And we had obviously 20 people, so we actually correlated landing position of the first eye movement with the fixation point of maximum performance. It was a correlation about 0.81, which is significant, but it was quite good. Now you'd be tempted, like we were, to just say, okay, now we know the answer. They're actually making an eye movement there because that optimizes their performance. So the system has calibrated. That would be great. We would love that. But we don't have the answer to that because we don't know what's the causality. Because the other possibility is the reason you actually are really good at when you fixate here is all your life you've looked at the noses. So if you from birth you actually look at noses and you do the task like that, well, perhaps that's why when you actually force to fixate the nose, you're really good, and you fixate on the eyes, you're not so good. So here we don't have the full answer. We don't know what the causality is. And because of this, Matt is actually doing more elementary measurements of these sorts of performance maps for very basic stimuli, not faces, like things like Gabors and stuff like that. These are things that we love in vision science, little signals. And the idea is to perhaps connect, if we can actually connect something that's having to do with, that, to really conclude that people are actually optimizing their eye movements based on properties of the visual system. But we're not there yet, so we don't know. So we don't know which one is causing what. Okay, now I don't have too much time, because I've spent so much time on this. So we're going to zoom through the second part of the talk. So that was the first part of the talk. You see some interesting data. I hope you agree. Not all the answers, but interesting data. Second part of the talk, we're going to try to see, well, can you actually infer from neural activity how people are going to be doing in these tasks, how well they're going to be doing. So just give me the one sentence on neuroscience for those of you that are not reading neuroscience. So sort of the debate whether, I know when I was a kid, there was people who would say, well, our thoughts and is cognition represented in the brain. For the most part, that debate is over. So most of things that we think of as cognition and perception and even emotions, the debate, it's represented in the brain. Now the question that is much harder is, how does the brain represent it? How to decode it? What is the code and how to get it out of there? And we're actually limited by sort of the tools that we have. You think, oh, we have lots of fancy tools. But just to give you an example, the visual cortex has about 140 million neurons. We cannot, well, firstly, we can't, for the most part, except for patients and exceptional cases, we can't even measure from single neurons because obviously that is a little bit invasive. So what we have is non-invasive techniques. And the one I'm going to be talking about, the first one is this is actually with Barry Giesbrecht in his lab. He's an expert at this. So I've learned this from him. And so this is EEG, or electroencephalography. And what this is just really measuring electrical activity at the level of the scalp. So that's pretty indirect. Because the neurons are in there, and so you're actually, this is sort of a new, so you have in this case 64 electrodes, 64 numbers, but you're sampling it through time, which has really good temporal resolution, about 512 hertz, and the idea that you're measuring is sort of groups of neurons that are actually synchronized in activity, currents there, but this is a dimensionality reduction, right? 140 million to 64, and that's just V1. So what we're going to be doing here is we're going to measure that activity while people are doing the task. So now this is a little different task. Here, the stimuli comes up really rapidly, 40 milliseconds, and I don't know if people can see, this is sort of low pass noise. Do people see anything here? No? Can somebody identify something here? Not the ones that seen this? Okay, now I'll tell you like a car, yeah, sort of is it like a station wagon or is it a car? Nobody sees a car there? What about a face here? Okay, face was okay. Car not so good. Okay, the car might be harder to see. So there's 12 different cars, 12 different faces. They're just presented. People have to say whether it's a car or a face, and they use a rating. And at the same time, we measure the neural activity. Are we going to do this? So what are we going to do with this? So people gave on each 12, people gave a behavioral response using this rating. So the rating just tells you how confident you are that it was a face, and how confident you were that it was a car, and for reasons that I'm not going to into, this is a good way to measure things. You can always collapse it into binary decision. And then the EG activity is going to be fed. It's an array of 64 numbers, but then about 512 hertz. And then we're going to be analyzing about 400 or 500 milliseconds of data. That's all fed to a multivariate pattern classifier or machine learning algorithm. I know people are familiar with that. Those that are not, I'm just going to describe the most basic one, just a sort of a linear discriminant, where you just get all your input variables and you do a weighted average, and just you come up with a single decision variable, which you're going to be making a decision based on that single decision variable. It's a car or face. And these things are trained in a group of training data sets, which is different from the testing set, because otherwise you bias it and all these things. But basically what we end up getting is a decision from the human and a decision from the classifier acting on the neural activity of the human. And then what we're going to do, here's what we want to try to do. If the person does really well here behaviorally, is it true that the classifier analyzing that person's neural activity is also going to be good? And if this person is really bad at this, is this classifier going to do really well? So is there a correlation? So here's our behavioral performance. Nothing to get excited, but just to show you that there's, even in this task, we still I've ordered them. Sometimes I order them. So I've ordered people around, so there's huge differences. Chance is 0.5, 1.0. There's huge variability here. There's a very, 40 milliseconds presented image, so it's not surprising. Some people cannot see a thing. Okay, so here comes the data slide. So here's behavioral performance. And here we're looking at the classifier performance for that person. That's the classifier based on the neural activity of that person. So here's a person that, so this is a prototypical example, somebody that did really well in the behavior, and their classifier did quite well, and so somebody that did poorly in both. And the correlation is about 0.7. That's statistically significant. Some people don't like correlations, so they like to see something a little bit more tangible. So we can run a little simple simulation that sort of illustrates a point. Let's suppose that I actually get the neural activity of two people, and I measure their pattern classifiers. So these are two randomly sampled people, and I want to predict from the neural activity which of these two people did better in their behavioral performance. So chance would be 50%, right? And we can assess, how well we can guess which of these two people from their neural activity whose behavior is actually better. And if we do that, it's still, it'd be nice to guess 1 out of 50 who's the best. So here we're just doing 1 out of 2, so we're starting easy. But the classifier, these are just standard techniques in EEG, which you don't have to be concerned. But we get about 85%, which is, not bad as a starting point. Now, when we got this, we thought, oh, this is a good result, and we got sort of interested. I'm going to skip this one, which is you can do this analysis through time and track where the information is through time, how it builds up. And this is a blank slide. It's purposely a blank slide. So when we did this result, we were like, we got, oh, this is a good result. But you know, one thing that we didn't like about that technique, that it requires to train this classifier in each individual. So you really have to get some data for that, for this to work, to train the classifier. And we really thought, oh, that'd be great if we can get a universal classifier. One thing that actually worked for everybody. That would be fantastic. And we got a little bit arrogant, so yeah, we're going to get this done. We're going to, you know, we worked on that, and it was a disaster. It didn't work at all. And you know, when we try to basically train on, so what we did is we tried to train on 19 people and get sort of average across everybody, and then test on one person. That didn't work, and it just really sort of told us, well, that sort of gave us the idea that, well, there's probably a lot of differences in the actual neural activity across people. The patterns of neural activity are not the same across people. So then we said, well, if that didn't work, then we can try the following, which is the opposite of what we were trying to do, which is, instead of having a universal classifier, if that doesn't work, that means that there are differences across people's neural activity. What if we actually get 500 milliseconds of neural activity from a random person, and rather than predict what they saw, we try to predict who that person is. So here's how it works. I know this is, requires a little bit of thinking. So I have so these people all were, you know, separately were looking at this image, and I get randomly sampled the neural activity of one trial. So about 500 milliseconds of data from this person, and then I send it to a pattern classifier, and now the pattern classifier is not trying to predict what they saw, but they're trying to predict who this person is. It's sort of like a biometric. And people are trying this around the world. And what we found is, these are two different algorithms, sort of support vector machines, so, you know, but these are two different methods, but we had the best algorithm was about, you know, above 92%. You know, chance is 5%. Well, that was pretty good. So that was pretty good. But here comes the sort of interesting science thing, which is the blue thing. You say, what is that blue thing? If we lose, so remember, when this is the neural activity after the image came up. If we lose the neural activity of each of people while they're sitting waiting for this image to come up, so that's what we call the pre-stimulus activity. So they're looking at that cross. If we use that neural activity and try to identify who it is, we're at chance. And we have no idea what this means. We think it's profound, but we have no idea what it is.
Huh? How do you try to identify who it is? You don't not compare it to the last one.
So we have a classifier that trains it and then tests it. So it is that you have to train, so the classifier has to see the activity of the person before. Yeah, if not, I would be in the Jay Leno show, if I could do it without training on. So you have to train the classifier to sort of, these are the features of this person, and then you get new activity from this person. And for the what? Oh yes, this is for the rest state. This is, and it doesn't work. That is the interesting thing. This is a classifier. So it's the same classifier, and it's just there's somehow what this is saying is one where a lot of people say that rest activity. The other thing is, well, you call this rest activity, but at least this time before there's somehow not enough signal that is different across people that the classifier can pick it up. So the classifier. So the classifier trained and tested for this time period, and there's a separate classifier that it's not this classifier working here. It tested in the pre stuff. So that would be obvious. Like if you test it trained here and then you test it here, there are different features. So no. So this is trained and tested in the pre-stimulus activity. Okay. So now. You'd say, okay, that's interesting. Now we also tried something that we have an expert visiting from Stanford in this, so we're just starting at this. During my sabbatical in UK, I sort of gathered some data, and Koel Das, who's a postdoc, actually really spent a lot of time analyzing this data. Same task, but rather than doing an EEG, this is now functional magnetic resonance imaging. It's a totally different way of actually measuring neural activity. It's looking at the ratio of oxygenated and deoxygenated blood. This has bad temporal resolution, but it gives you the good spatial resolution that the other technique doesn't have. This sort of stuff here that you see is that stuff that Brian has pioneered, and I'm still learning with Tim, who came from Zoe's lab and knows all about this, which is a way to actually put stimuli and actually functionally segregate and segment these different areas that we know about, or that we're learning that we know about a lot in the monkeys, and we've in the last sort of decade and a half learned in humans. So we tried exactly the same thing that we did in the EEG and fMRI, and this is the sort of same plot, and we got 0.73 correlation. So that's sort of actually pretty similar to what we got in EEG, although this is a totally different technique. So we thought that was interesting. And then the only thing that you can actually do in fMRI you can't do in EEG is sort of run the classifier on separate areas. So these are different functional areas. All these, V1 is the primary visual cortex. So all these mostly have to do with form perception. And I try to get updated. V4 used to be the classic color. Now I'm reading the V8 is involved in color too. And the LO is lateral occipital. That's related to, established that's related to object. And FFA is a fusiform face area. And this is the correlation coefficient between, so basically this is the number, you could actually plot this, behavioral versus classifier performance, but the classifier is running on the voxels of each of these areas. And this is...
Those errors are 1 sigma statistical errors?
These are standard errors, I believe. So all this stuff, I think right now, most of this is like you say, you believe, not significant, except for this guy here. This guy is the thing. So we've done the statistics. All these guys are, yeah. You know how many, and these are 1,000 trials, so which means that people were there suffering. So this is tough business. I wish we just did theoretical stuff, but this is like a lot of work. You have to be there in the lab. So and being in the magnet, I invite you to go in there someday. But it's a good observations. But
that's because those plots you showed before the correlation plots are pretty scattered.
Well, this is on the whole. Well, it's statistically, if you ran one of these, theoretically the correlation coefficient that's significant is about 0.44 for this. But now, once you start doing many, you have multiple comparisons, so you have to do a correction for that, and so on. So now, once I do this per area, this is because, like you say, these guys are very scattered. If I plotted this one, these would be less scattered right there. But the yellow does the best. Is not surprising because we did have objects in there. Sort of the FFA, which we had faces also in the image, is a little bit more poor. That's a little bit surprising. But this in general is actually not surprising. Now, believe me or not, this is not that easy to do. So people have tried to just correlate actual behavior with the areas. And this is actually sort of the rank order simulation. So we can skip it in the interest of time, but that just tells you that there's stuff there. So the fMRI gives us less surprising results, but it's converging with the EEG that we're able to. So it's sort of a miracle. We're able to tap in from this very indirect measure. We're able to get stuff out that is actually related to behavior, even though the methods are expensive and are very technically impressive. But but we're still there are 140 million neurons, and we're doing these basic measurements. So. Now you'd say, I think you're asking, well, how is this? Will this work for something really realistic, a real task? Because again, there's this car, faces, and here this is data from a recent paper by Steve Engel, and he's not using pattern classifiers, but he did the same thing. He put people in the magnet, and he actually had doctors read these chest X-rays, and he actually measured bold response. For these different images for different people. Some of these were actually sort of trained radiologists or more experienced radiologists, are the ones who were novices. And what he got is that in general, so the red ones actually are the novices, and the green ones are actually the experienced radiologists. And this is not pattern classifier performance. They were using something that was a little bit more basic, just a change in MR signal. But he got and this is a measure of performance, a significant correlation between the percent MR signal change and the performance for the experts, but not for the novices, which is sort of interesting. The novices, that there was in red, there didn't seem to be much of a correlation. So we're hoping that we're actually been talking to Steve of actually trying to combine forces and try to move in this direction with radiologists and use some of these classifier techniques that we've developed to try to see how whether we can actually track performance of not just with our undergrads or grad students, but with these more realistic, challenging tasks. Okay, so sort of, so as it, I'm on time. So it's impossible to come. We've covered so much that it's like, how possibly can I? There is no grand theory. There is no concluding remark in terms of a single remark. But I want to bring back to the starting point where we had these challenging tasks. And I hope that you're convinced that, and maybe you're going to check out the NPR little clip this morning about called Guns, Tumors, and the Limits of the Eye that just happened to come out today, the day of the talk, and a friend of mine said, "Oh, this is what's out." That there are this is human performance in these tasks are a real problem. There are variations in how people interact with images. Some of these we sort of have an idea that we can explain in terms of training, whether people are experts and novices, and there are other ones that have to do with just individual differences, and we're just scratching the surface of trying to understand what are the different reasons why people perform differently with these tasks, and why it is that they interact with the images differently. So, thank you.
Just have one question or correction. You kept referring to 140 million neurons.
It's an estimate.
Aren't there 100 billion neurons in the brain?
V1, visual cortex. V1. And I didn't put the error bar, which is, I know Brian knows, it's what, I've looked it up a few times, and there's like different estimates of 140 plus minus 100. There you go.
In this EEG, you said
well, we're measuring for many more, right.
like 100 billion.
That's right. It just happens that a lot of the information happens to come from the sort of this area, which is more than V1. So yeah. So what would be a good estimate for like all the visual areas together? If I don't want to put in, what is it?
5 billion.
Billion? 5 billion. There you go. So that's for all the visual areas together.
Marty?
If you said this, I missed it, but do the people whose eyes tend to focus on the nose area perform better or worse than the people who tend to focus?
All these good questions. So we've really wanted to do that. So we thought if the first, when we saw the variability, we thought, oh, the closer you are to the sort of optimal observer, closer to the eye, that would maybe correlate with performance. And those that were looking at the nose are doing worse. And it's not necessarily true. So we tried, and it would be a great story, but it doesn't pan out. And those people, could be better than the ones looking at the eyes.
We have a question back.
Yeah, so I study genetics of complex traits in model organisms. And so when you say that this varies, I'm not at all surprised, because if we measure any behavior or any trait in any organism ever, it always varies. And the question is always, whether it's due to genetics or experience or state. So is there a way to put all that into a model together, right? How are you going to ever break it down into different potential explanations?
If I started my son studying genetics, maybe if it was a general and put him, maybe he would. I don't think I'm going to be able to break it. I mean, I wish I could, but that's a fantastic question. I don't have to. I'm not even a geneticist, so I'm not. It's really beyond my any capabilities I might have. I would have to go back to school to be able to answer. I could. It's a. But. Well, I've tried to break it down as much as I could in terms of expertise by starting people with a new task or trying to bring them back to make sure that it's not a bad night's sleep. But we're at the point. This is so basic that we're at the point of trying to get people in society to recognize that people are because I showed you some challenging tasks to recognize that there are people that are good at this and people that are bad at this. So we're really in a sort of very early. I mean, I wish I wish I could answer those questions or had the time and the IQ to answer those questions and the knowledge. But I mean, that is a great thing. And I think for more basic things, and people are starting to do, even in our department, genetics with combining genetics with neuroimaging. So that is something that's starting. But I haven't thought about it, and I would have to give it a lot of thought if you could actually connect sort of some genetic signature with these different levels of performance. I mean, it'd be great if yeah.
The question over here. Were there any cases, this is your novice versus expert, were there any cases where your classifier was doing better could do better than the person? In some sense, the information is there, the person is making the decision wrong.
Yeah, so it was a very good question. So I should have said that part of the EEG, the reason I got into this part of the EEG is because I saw this paper by a Columbia group in biomedical engineering. They actually have better results than we have, and they have their classifier does a little bit worse than the behavior. So if you look at their neural activity, and we typically do worse. Well, for a bunch of individuals, and now we're doing other tests, we can get close, but we have not got better. People measuring single-cell neurons will report through history, the last 20 years, that they picked up a neuron that knows more about the task than the monkey itself. So that's been done in single cells. That there's, which I was in graduate school, in my graduate course, I said, well, there's, when you connect your little PDA to your neuron and forget what your impression is, just tell get your expert neuron to do the task. But that's a little bit, that's another one that I'm not going to work on.
Okay, the next question from Walter.
Yes. There is a very simple and widely used tool in this broad area of trying to understand vision, distortion, perception. And I didn't hear you mention it, and I wonder whether you or other people working use it. It's the so-called Amsler grid.
The which grid?
Amsler, that's the name, A-M-S-L-E-R.
I'm not familiar with it. We have people that are more experts in it, so we have maybe John and Brian. Have you guys heard of it? Give me a little hand.
I'm sure. And it's particular interesting stimulus has some illusions associated with it. But where are you headed? I just wanted to hear more about the question.
It has a different character from your faces that you use for your subject. It's a simple square grid, and we have used it and find it very, very useful to quantitatively organize the data.
Amsler grid.
Amsler.
I looked it up. I know the Herman grid, but that's a different one.
For the technologists and engineers in the room and the tool makers, if you have could you show us your wish list of what you would like to be able to measure if you had your wildest dream?
I get scary, but my wildest dreams. So what sorts of things you would... well, there's something that Barry is trying. We have a new grant that is a little bit more applied. And one thing that we've tried here, because we tried, Coel tries. Hey, Coel, let's try this. And then I come, so she works day and night doing different things. So what we've tried that we would think would be very useful is, and we tried it here, can you actually predict whether somebody's going to make a mistake before can you actually predict before the stimulus comes up? So before the stimulus comes up, if you look at the neural activity, can you actually predict whether somebody's going to make a mistake or not? That would be very useful, because that would be you would be able to you'll be able to basically have an idea, an independent measure of whether somebody, and the idea there, this would work if you had some neural activity that was, if you're daydreaming or something like that, so if you're just not on task and your attention is super diffuse, if that actually has some signature that's reproducible and systematic. And typically when people are daydreaming and something comes up fast, they typically do a mistake. So these are the things that I think would be very, very, one of the things that'd be very, very, very useful. If you can figure out from neural activity that somebody is about to do a mistake. I mean, with this we can actually predict choices, which I haven't talked much about, but I have the data in the next few slides. But so that's one thing that would be useful. I think obviously improving all the stuff that we have, I think it's nice right now, but it's not up for getting a product.
I'd like to ask you a question. Let's go back to the eye movement. When you talked about people that focus on the eye and people that focus on the nose.
Yep.
People ask you whether there's a correlation with gender. There are obviously many other things you can ask to correlate with intelligence, age, race.
You can do. Oh yeah. So in fact there is a paper that is. I guess nobody knows the paper, so I can say it's not a great paper. That actually is looking at across cultures. Actually, they're using, somebody help me, East Asians versus something like that, and they're actually they're seeing small differences in where they're looking because of that.
There's a book about looking at some fish tank where some races focus.
There's some evidence for that, but I don't. The stuff that's out there, I'm not super convinced. I mean, I'm barely convinced by my data.
Well, what I'm thinking is that when someone looks at someone's nose, all of the rest of the information is still there. Like, all of the rest of the eye information is still being recorded in their. I mean, they could still see it in their brain. So I'm thinking it shouldn't matter where they look, because they could still be mentally focusing on their eyes while looking at their nose. So
that's a good point, but so.
that should really make such a big difference.
So remember, I'm not talking. I won't tell you about what we're working. Another model we're working, but the idea is that is partially true, but not true, because if you remember that you have a foveated visual system where the center is in high resolution. So it's like if you just vision scientists cover your ears. It says if you have a 50 megapixel camera here, and then in the periphery you have a cell phone. Well, not an iPhone, my cell phone, like a really bad, those. So if you actually look at the nose. It is true, you still have the periphery, but you are limited in how much information because of the fact that your resolution is going lowering as you go toward the periphery. Now, I'm not going to too much detail, but you actually can build into this ideal observer this fovea, and we call it the foveated ideal observer, and we're working on it. And what will that do is it will bring those. When you have a fovea, it will bring the optimal point of fixation from the eyes a little bit further down, and we're trying to make that prediction because. The idea there is that you're trying to really get some, with some information in the nose and in the mouth. So you might want to just hit there. So you actually, so your peak of resolution is there, but you sort of get the eyes and you get the nose. So, but we don't have the whole story yet. So.
Just following up on this question, if you change the level of illumination, then the depth of field would change, right? So it will see whether they're focusing a lot more closely or not.
So there's going to be
some depth of field where the thing is going to be sharp.
Right. So.
When you're focusing on the nose and I change the illumination level, you could see whether you're sharply focusing.
So you're talking about the depth of field. You're talking about the depth. Hold on. You're not talking about the depth of field of the actual picture taken. So you're talking about where you focus and the changes in illumination. So the luminance of the faces, then there's noise, and it is true that where which we haven't investigated yet, if you make the task easier by actually making these faces higher contrast, it might be that the people will actually fixate on different points. And let me just take it to the extreme example. If the noise is not very limited and it's a very easy task, you might not even have to make an eye movement. You might just stay where you initially say. You can perfectly see the faces. If the faces are sufficiently different, you might not need to actually make an eye movement. That doesn't mean that people won't. So there might be, like you say, if you change the luminance and the signal-to-noise ratio, even this Bayesian ideal observer might either change its prediction or actually have, there's less cost of actually not performing the best eye movement. So and that might actually modulate human behavior, and we don't know.
David?
So one point I saw lecture with lots of fMRI, and people were trying to discover how people thought, and often averaging over observers like you are. And what I was struck at was how different individuals are, how the variation from individual to individual.
Walter was saying that nobody could hear you, so maybe you could say a little louder.
I could hear it, but nobody in the back.
Yeah, so.
I was just remarking that when I saw once in a lecture people trying to average over fMRI observations and figure out how people were locally, I was struck by the enormous variation from individual to individual. So I thought that, and that wasn't determined by genetics, but perhaps by development, because the brain, after all, develops, learns in its interaction with the world. When it comes up with a solution, like you look at noses, or you look at eyes, to recognize faces, it sort of stops its development. It doesn't necessarily perfect beyond finding a solution. Maybe not upward. And that it seems to me that one should try also to look for many different ways. Now you seem to have identified two
which might be in this limited task you're describing of recognizing faces with one eye movement, look at the nose or look at the mouth. There might be, but you're also there averaging to a large extent.
Okay, so he.
There might be. So I guess the question is more: is that a reasonable point of view that there are indeed, even say among identical twins, might be very different ways of thinking depending on random choices that, depending on the history of their own brain development and the fact that they found the brain in its development found a solution, might not be optimal, and some other brain will find a different solution, even with the same genetic starting point, and that you should actually be looking for a whole different patterns, which are possible fixed points of various human brain development. You should be expecting to find maybe 10 different strategies that would be hit upon.
So
One or two. Why are you trying to look for the optimal solution? Well, the brain, once it finds a solution, presumably doesn't spend a lot of energy in its development finding the optimal solution.
So okay, so if I got so there's a few things. So the methodologically, actually, I'm averaging the classifiers, but actually here, all of these analyses are actually done on an individual basis, and that's what Brian Wendell and Marty Sereno, these people in Vision, actually pioneered, and actually it's different from a lot of the rest of cognitive neuroscience. So these this segmentation for this person is totally, what you do is you find these areas for each individual person, and if you look at this person, it's going to be different than another person, it's not going to be the same. So the analysis is all tailored individually. Now the bigger question, about optimality. So I'm going to confess, there's sort of a little cult going on in our field, which I am part of. It's sort of laughed at when I was in grad school, sort of, but I was lucky. It sort of became a little bit more fashionable later, and that's why I got a faculty position. And what we believe is, in fact, that some of how people behave are, in fact, not always optimal, but in fact that you actually do things that are semi-optimal because these things, vision is important to survive. It was, at least. Now it's a little bit less important, but it was, and finding food and not being and it's not just it's not just perceptual psychology and vision. I mean, if you look at biology and vision, there's tons of these explanations that tries to that evolution and also through learning. So the two things, there's the sort of the evolution has given us sort of a good solution, which perhaps is not the optimal, but it's a good solution, and then through learning you also become quite, not always, but you get quite good, and that using these analyses stuff in terms of how an optimal or rational observer would do a task gives you some idea of an explanation of why humans sort of behave this way. So that's.
I guess the point was, if you believe that, that's a point of view. If you believe that there might be 10 equally good solutions,
okay,
you would treat your data very differently than if you are trying to find evidence for or against.
I got you. I think I got you now. Okay, I think I got you. So let's go back. So do you remember I told you about the fovea that ideal observer? This is like this. I'm putting a fovea. I'm sort of the properties of how performance degrades as a function of. So what we're trying to. You can do that, and actually, but you can do measurements on these individual people. What we're trying to do, and actually tailor this optimal solution for each individual. So, given that there's information in the images, and you're stuck with this, how performance varies as a function of eccentricity, that you're stuck with your visual system, what would be the optimal eye movement? And that's going to give you a different solution for each person, if indeed people's performance as a function of eccentricity varies across individuals.
Perhaps a couple more questions. There's one in the back.
Going ahead with this question of learning and development, if you actually presented faces, artificial faces with keeping their eyes constant and changing their nose, would then the person who looked at the eye do worse than the person who looked at the nose?
That is in my grant that is actually being was reviewed yesterday at the NIH. I don't know what the score is. I don't want to look it up, get depressed before coming here. But exactly what we're doing is we're actually not even using artificial faces. We got a Photoshop expert, and a Bavarian, and they worked on it for a year of getting the actual faces we have and getting a common face and just manipulating the nose. So now we have a data set that just has the nose has the information, or the same data set with just the eyes. So we're able to artificially put and we haven't got what you're asking, but what we got is we started with somebody that's an eye person looking at the eyes. We gave them that, didn't tell them anything. And then we looked. Well, how do you think the eye movements start going? Zoom. And that's it. Yeah.
In terms of the problem on its head, the hypothesis that the eyes are designed to help us discriminate between the eyes, the way they're physically from the outside, to tell other people who you are. Okay, so what coincidence? Why is it that the place that gives you the most information is also the sense organ? And I could look at people's hands.
I never thought about it. It's a fascinating one. I have to give it some thought. But it sounds, I wonder if there's some counter example there, right? Because I mean is that right? But aren't there animals that identify themselves by other parts of their body? I think there are, including buttocks.
Okay, I think a lot of people are left from cookies who are invited to let's thank you. Thanks. That was fun.